And with that, I'd like to introduce Russ Poldrack, who's at Stanford University. Russ has been at every one of the NeuroHackademys and NeuroHack Weeks that we've done here, and we're really grateful for that, and it's always been great. Last year when we talked about this, he was no longer planning to fly, and he thought he might do a road trip. Ironically, no one is traveling, and he's not traveling either. And but we're really grateful that he's showing up here on Zoom, and he will be talking about reproducibility in fMRI and what the problem is. Thanks, Russ.
All right. Well, thanks very much for the invitation. And sorry we can't all be there in person, but I am happy to see how many people are able to join remotely. Let me share my slides. So are you seeing slides or presenter view?
Slides.
Okay, great. Cool. All right. Well, so I want to talk about kind of, why is it that we need to care about reproducibility, particularly in fMRI, but, a lot of the issues that I'm going to raise are really sort of more general to lots of other types of neuroscience data as well. So I'm going to start, by talking about, how much resources are actually being put into research using fMRI. So I just did a search the other day on the NIH website for grants in the 2020 fiscal year that had the term fMRI in the title or abstract, and found 1,200 grants. And if you add up the money that goes into those grants, it's almost $500 million. Now, obviously, some of them might just be mentioning fMRI, but a lot of that is actually, research that's collecting fMRI data. And just did a quick and dirty word cloud of, kind of what are these grants about? If you look at the abstract, and clearly there's a lot of interest in understanding the mechanisms of clinical disorders, effects of treatment and change and intervention, effects on development, coming up with, predictive biomarkers, all these sorts of things, right? So clearly, there's a faith on the part of at least, funding agencies that we could use fMRI to actually tell us something useful about these questions. So let's just look at, one particular question that you would hope fMRI would be able to answer, which is, what's the brain dysfunction associated with major depression? And I'm showing here a figure from a paper a few years ago by Mueller et al. In JAMA Psychiatry. This is kind of a meta-analysis of meta-analyses where each of the dots here is showing a result from a previous meta-analysis of differences in brain activity on task fMRI studies for either emotional or cognitive tasks, comparing depressed individuals with healthy individuals. And each of these previous meta-analyses had shown, consistent differences across studies between depressed and healthy individuals in brain activity. Now, Mueller et al. Kind of reanalyzed the papers that went into each of those meta-analyses, a total of 99 published studies. And they found that when you do the meta-analyses the proper way and put all these data together, they find no convergence across these 99 studies. So there's 99 published studies, each of which claim to have found differences between depressed and healthy individuals, but altogether, they basically come together to tell us there's no convergent, consistent difference. And this isn't the only example of failures to either replicate or find consistent results across imaging studies. This is another one published a number of years ago that tried to replicate a number of brain behavior correlations, and basically found that across these 17 different effects, there was really none of them that's kind of solidly replicated the original significant finding. So this is all a little bit scary, right? There's at least a canary in the coal mine suggesting that that we have serious problems in our ability to come up with reproducible results. And so the question is, how can we fix it? And so a few years ago at Stanford, we started a new center called the Center for Reproducible Neuroscience that is sort of aimed at coming up with ways to try to address these issues head on. And so I'm going to talk about, at various points, some of the work that we've been involved in today. One of the major efforts is around open data, basically, helping provide tools to let people share different types of data, including Open Neuro and NeuroVault. And we've also been collaborating with Tal Yarkoni on NeuroSynth. We've developed a number of open tools like MRI-QC and fMRI Prep. We've also been involved in developing open standards, and I'll say a little bit about BIDS later on, which is a standard for organizing shared data. And finally, I'm going to talk a little bit later about some of the meta science work that we've been doing, particularly around trying to understand kind of the effects of analytic variability, in the wild. So I like to think about what we're doing in terms of a design problem. So if I was in the room with you all, I would ask you if any of you knew what this was. And sometimes somebody in the room will know that this is a commercial paper cutter. And you notice that the paper cutter has two big green buttons on it. And those buttons are there to basically prevent people from doing a dumb thing that people would often do with a paper cutter, which is to stick their hand in and hold the paper, and then potentially have their fingers cut off by this very large blade. And so these two green buttons, you have to press them down, and then you hit the foot pedal to cut the paper. And this is basically what designers do all the time, right? They try to build systems to afford the things that we want people to do and disafford the behaviors that we want to prevent. Now, one of the things we have to worry about in terms of research reproducibility is the fact that humans are really good at fooling themselves, at self-deceptions. And so one way to think about, what we're trying to do here is kind of push people and design situations to drive people to kind of, work against their own worst instincts. And another way to think about this is in terms of the concept of choice architecture from behavioral economics. And the idea here is that, anytime we go into a situation where we have to make decisions, there's a set of features that drive us either toward or away from particular choices. One of the obvious examples is defaults, right? If you go into a neuroimaging software package and it has a default smoothing kernel of 5 millimeters, you're very likely to just use that default, right? And so sometimes those defaults can be good ones, and sometimes they can be not so good ones. And so the idea behind nudges is basically kind of designing a system to push people to do things that we think we want them to do, even though they always have the option to do other things. We're not limiting their freedom, but we're, for example, if there's a particular default that we think is the right one to use, then we make that the default rather than something else. And we also sort of think about the kinds of errors that people will make and try to expect and prevent those errors. So many of you may be familiar with this paper by John Ioannidis, Why Most Published Research Findings Are False, really kind of one of the bellwether papers in the whole reproducibility crisis. And in this paper, John laid out a number of features that he claimed were associated with kind of the likelihood of true findings. And so one claim he made was the smaller the studies in a field, the less likely the findings are to be true. The second is the greater the number and the lesser selection of tested relationships, that's, how many different tests you're doing, the less likely they are to be true. And third is the greater the flexibility in designs, definitions, outcomes, and analytical modes, the less likely they are to be true. And clearly all of these are, are sort of indicative of what happens in fMRI research. So I want to focus first on the question of sample size. Now we know from a number of studies, or a number of meta-analyses, that research basically across biomedical science is generally underpowered, and we know that neuroscience is no different. The left panel shows the results from the power failure paper by Kate Button and colleagues, where they looked across both structural imaging papers and rodent papers and showed that the majority of studies that they saw were badly underpowered. And the study on the right is a follow-up of Marcus Munifo and colleagues that basically shows something very similar, that most studies are badly powered to find the kind of the median effect size that's been reported in the literature. If a study has, here we see, in both cases, many studies have 10% power or less. That means that even if there is a significant effect of a particular size, 90% of the time, your study is not going to find it. In medical research, this is often referred to as a futile study. And you would never be able to sort of run a clinical trial with that low degree of power. Now, you might think that, well, that's fine because we're controlling our type 1 error. And so, we can run a bunch of studies, and if we're underpowered, that's fine. We'll occasionally find an effect, and we're controlling our type 1 error. So our overall rate of false positives will be, as expected. And that's true, but Ioannidis pointed out that may not be the number we really care about. And argued that what we really care about is what he called, or what statisticians call, the positive predictive value, which is the probability that any particular positive result is true. So rather than conditionalizing across all findings, we conditionalize across all positive findings, all significant findings. And the point here is that basically when you have low power, even if you find a significant finding, it's very likely to be a false positive. So here's a thought experiment. Let's say you're a physicist and you're doing a study, but unbeknownst to you, your detector is broken. So it's just giving you white noise. And so let's say you run 100 experiments and you're going to get 5 of those giving you significant findings. So your false positive rate is still 5%. But your positive predictive value is 0, right? None of those is a true finding because they're all just generated by noise. And then as you fix your detector, the likelihood of actually getting a true finding goes up, so your positive predictive value goes up. And so the point there is that, with low power, most positive findings, which are the ones that are going to get reported in the literature, are going to be false. And we'll see examples of that in a little bit. Another point is that, if you do find a significant finding in an underpowered study, this is what we see on the right, that the effect size that you estimate is much larger, is biased upward from the true effect size. So here's an example. Felix Schomburg in the blog post at the bottom pointed out another problem that's sort of obvious when you think about it, but even I was kind of struck with the magnitude of this effect, which is that with small samples, your statistical estimates become really unstable. So what I'm showing here, these are actually real data from the NHANES data set, which is a large health data set in the US. And I just basically, each of the lines here is basically one sampling run where I just randomly took samples of increasing size. And for each sample, computed the correlation between body mass index and this measure of blood sugar, HbA1c. Now we know for the full sample, which is about 6,000 people, that correlation is 0.21. Highly significant, not a huge correlation, but definitely something that's real. But this shows you basically the red lines here are the 95% confidence interval for the correlations across 1,000 samples, right? Each of these little sort of thin lines being one sample. And basically what you see here, Felix came up with this idea, what he called the corridor of stability. This is the dashed lines show you basically, this is the true correlation plus or minus 10%, right? So the idea is like, you'd like to be pretty sure that, when you collect a sample, that your observed estimate is going to be within, say, 10% of the true estimate. And what this shows is that, even with a sample size of 100, the 95% confidence interval is way outside that 10% corridor of stability. So you get, even with samples that we think of as being reasonably large, like 100, the estimates can be kind of wildly unstable. So how does this affect things in practice? Well, I think a great example comes from the study of candidate gene association. So back around 2000, people started publishing, when it became possible to genotype individuals for particular genetic variants, you started seeing a bunch of papers claiming to find associations between particular brain measures and particular genetic variants. So this is a meta-analysis of associations that were reported between a particular genetic variant in the BDNF gene and the volume of the hippocampus. So the plot I'm showing at the bottom this is taken from this Molenbeek review, shows on the y-axis the effect size that was reported in the paper in Cohen's D. the x-axis is the year of publication, and then the size of the dot is the reflective of the sample size. And so what you see early on is that you have some very large effects being reported, generally from very small samples. Over time, as the samples get larger, the effects basically start getting smaller, and ultimately the effects tend down towards zero. And in fact, we know now from very large genome-wide association studies that, you know, when you have samples of 20,000 individuals, none of the genetic associations that have been reported in these previous studies, like BDNF and a number of others, actually replicate once you have enough data. There are other genetic associations that actually do replicate, but none of the candidates that people had studied early on were there. And so the idea is, you know, these small studies led to very unstable estimates. And, you know, with publication bias, you know, anytime anybody finds a significant effect, they're going to publish it. You don't see all of the many non-significant effects. And in fact, I remember back around 2003, 2004, going to talk with people at various universities and asking them, everybody was interested in this COMT association. And I would ask people, what are they up to? And they say, oh, we've been looking at effects of COMT. We don't really find anything. You know, a number of people told me that they were basically not finding anything. Those null effects just went into a file drawer and really didn't see the light of day until later on. So how are we doing at fMRI? So this is now a slightly outdated meta-analysis. It's from a few years ago. And so it gave us data up through 2015. And in 2015, the median fMRI study, the median sample size was 28.5. And using some assumptions about the nature of the effect that you can detect with that sample size, the median study was powered to find an activation with a Cohen's d of about 0.75. So the question is that a reasonable effect? We want to make sure that we're powered to find reasonable effects. The problem is you can't just take published effect size estimates, because they're going to be biased because they were obtained by thresholding the whole brain. And so we tried to estimate realistic effect sizes by basically taking the Human Connectome Project data and then using the prior regions of interest that they had given us, along with maps from NeuroSynth, to basically say, where are the voxels that we're really sure there should be activation for this particular task, like a motor task. And then we can look just in those voxels to get an unbiased effect size estimate. And when we do that, on the right here, you see the Cohen's d. On the left, you see the percent signal change. This is kind of the average size of the within subject effect. You know, how much does the signal go up when they do the task? And you can see these are, like, you would rarely expect an average effect size more than about 4% in fMRI data. And in places like motor cortex, you see 3-4%. You see pretty large effects even in amygdala for like looking at emotional faces. But what we really care about is, you know, the size of the effect with respect to the variability between subjects. And what we see here is that, you know, we see some tasks that have effect sizes of about 0.75. Those are kind of, you know, things like moving your hands in motor cortex. So kind of like the biggest effect that we would ever expect to see is seems to be on the order of about 0.75. I mean, that suggests that, you know, for kind of realistic, interesting effects, we're underpowered. So there's a question I see: what is effect size? And so effect size here is, so for example, Cohen's d is basically, let's say that, you know, you want to know about what's the Cohen's d for activity in the motor cortex when somebody moves their hand compared to when they're doing nothing. So what you would do is you would compute for each individual let's say the percent signal change. How much does the fMRI signal go up within each individual when they do that task compared to doing nothing? And then you would take across subjects, you would ask basically, what's the mean effect size in comparison to the standard deviation? And so it's basically a measure of how big is the effect with respect to the variability across individuals. Hopefully that's a helpful answer. So let's look at the data from that Mueller et al. Study. This is plotting on the y-axis sample size and on the x-axis, the year of publication. And I've colored in anything in red any sample size with a particular group with less than 20 subjects per group, I've colored in red. And that's based on this argument from Simmons et al. That any sample smaller than 20 per cell is simply not powerful enough to detect most effects. And so there's usually no good reason to decide in advance to collect such a small number of observations, right? So the idea is that what's probably happening is that people are collecting some data and kind of looking at the effects as they go along. And then when they find a significant effect, stopping. That's what they mean by a flexible termination rule. So we see that, presumably one reason for those, unstable effects in that Mueller et al. Meta-analysis is the fact that so many of these studies have, what I think is now agreed to be grossly insufficient sample sizes. So another, I think, more striking way to frame this is that 634 individuals, that's the number of depressed people in those studies, volunteered to come get in an MRI scanner for studies that were almost certain to generate false results due to insufficient power. So a question cell means group in this. So when I say per cell, what I mean here is like per cells would be like number of depressed individuals, number of healthy individuals. Another way to frame that, if you're worried about, climate change, is that, by some, obviously you have to sort of come up with ways to estimate the amount of CO2 generated by MRI scanners, but, hundreds of tons of CO2 equivalent to, many cars driving for a year. So, that's what happens in kind of standard, statistical testing. What about, many people now care about coming up with predictive models to actually, predict something, come up with, for example, a quote unquote biomarker for a disease. And what we now know is that, these issues have similar impact on those types of analyses as well. These are simulation results from Gael. You're going to hear more from Gael later in the week. These are simulation results from a great paper that Gael wrote a few years ago called cross-validation failure, which basically showed that with small sample sizes, you get highly variable estimates of predictive accuracy. This is a real world example that I created where I took, again, data from that NHANES data set and asked, I want to predict whether somebody is a smoker or not. And I used just 3 variables: age, gender, and then one of the measures in that data set is how much cadmium, which is a mineral that is associated, it's higher in smokers, how much cadmium is in their blood. And again, in the full sample of 5,200 people, we know you can predict with about 70% accuracy, that's balanced accuracy, whether somebody's a smoker or not. But I looked again at what happens when you take smaller samples, and this shows you the range of variability, the range of accuracy estimates you get when you have very small samples, starting with 20. But look at, let's say even up to 80 subjects, you can find some samples that have 100% accuracy and others that have basically even below chance accuracy, right? And with very small samples, it gets really bad. And in fact, Gael showed in the literature that this is exactly what happens, that as sample sizes get bigger, the overall estimates of predictive accuracy for lots of different outcomes, lots of different, psychiatric disorders and so on, basically go down, and you see some people reporting 100% accuracy for particular predictive outcomes. I think that if you're seeing 100% accuracy for almost anything, you should be really worried, because that's really never plausible, and certainly with respect to psychiatric diagnoses. Okay, so a problem that's often raised is, that's great, right? Thank you for telling us how hard it is to do well-powered science, but I don't have, as an early career researcher, I don't necessarily have the resources to do a large study, right? To do the kind of study that I want to do. And so what should I do? And I think that the answer isn't to, go do an underpowered study. So I wrote a paper last year, opinion piece in Neuron called The Costs of Reproducibility, that tried to address this question of what do you do when you can't do the kind of study that you need to do. And I think one great quote is the one from Nancy Kanwisher, which is, if you can't answer the question you love the question you can, right? Find a question that you can answer well, and that could be using, existing data that are shared. That could be via collaborating with somebody else. That could also be kind of turning to focus more on theory or computational modeling rather than necessarily going out and collecting a new data set. And I just want to highlight, one example of a success story. This is Max Shine, who was a postdoc in my lab for a couple years, now a faculty member at University of Sydney. He collected no new data in those 2 years in my lab, but nonetheless was able to publish a number of high impact and I think really interesting, cool papers, all based on shared data. Some of which was, data that were available from, sites like the Human Connectome Project. Others, based on data that we sort of, got through collaborations. The point being that, there are options if you want to, do interesting science using these large shared databases. We've been particularly involved in developing the Open Neuro database, which now shares a number of different types of data: MRI, MEG, EEG, intracranial EEG, and now just hit 400 data sets, has over 1,200 participants worth of data. Lots of really interesting data sets there. So I think that if you have a question that you're interested in answering, looking there to see, is there one or more data sets that could help you answer that, is a good place to start. And obviously there's a number of other databases as well that have interesting data available. Okay, so now let's look at another set of issues that Ioannidis raised around the number of tests that we do and the flexibility in our designs and analysis processes. This is from that same review a few years ago, where we went through and asked, this is just looking at FSL. How many different analysis workflows could one come up with, just like choosing kind of all reasonable options for, for the different things? Like, these are all options that no reviewer would gripe about, I think, and often wouldn't even be mentioned in the paper. And we came up easily with over 69,000 different workflows. And in fact, Josh Karp did a review a while ago, published back in 2012, that actually looked at papers and showed that indeed there's a wide range of different options that are used in many different papers, from the degree of smoothing to thresholds and various other things. He also showed in another paper that this flexibility actually has an impact on results. So here he took a data set from the Open Neuro database, and he ran it through almost 7,000 different analysis pipelines. And on the left, you see the average activation, which this was one of our data sets. The average activation looks roughly like what we had published, fortunately. But if the really striking finding is on the right, which is the range of statistics from highest to lowest across those 6,900 pipelines. And what you see is that there are differences of up to 8 standard deviations between the highest and the lowest Z statistic, showing that, indeed these different choices can make a huge difference in the reported results. And we now know also that machine learning can make this even worse. This is a great paper by Brad Weibel's group called "I Tried a Bunch of Things," which basically showed that by just by doing things that seem innocuous, changing hyperparameters, you can actually get seemingly significant predictive accuracy from basically noise. It's also worth pointing out that there's nothing special about fMRI here. Another great paper from Steve Luck and Nicholas Gaspelin showed that there's a lot of things that are commonly done in ERP studies that can lead to very high rates of significant, but what they call bogus effects. So I think one thing that we really need to think about is moving towards pre-registration. Obviously, pre-registration for fMRI is hard because there's so many different things one has to do, and especially analyses. You might have an analysis plan, but it's rarely going to, not change with once you actually see the data. I think nonetheless, registering an analysis plan prior to accessing the data, and preferably, actually simulating data and coming up with all the analysis code prior to actually touching the real data. That's kind of the holy grail. Even my lab hasn't done that yet, but it's something that we're working towards. A couple of things to point out. One, just there's no reason that you can't, if it turns out that something you pre-registered turns out to be a problem, you can always do things differently. You just have to be clear in the paper that you basically modified, you kind of violated the pre-registration for particular reasons, and as long as they're justified, every paper we've ever published that has been pre-registered has had to deviate from the pre-registration in one way or another, and as long as you motivate it and justify it, it hasn't been a problem for us. It also doesn't prevent exploratory analysis. What it does is it makes it clear to you, kind of, it's easy to fool yourself into thinking that what you're doing was really planned when it was actually exploratory. Okay, so there's a question. Can you explain a little more what the term registration means here? Basically, what I mean is going to, there's several different websites you can use. One common one is this open science framework that you see here on the right. The idea is basically before you ever collect your data or analyze any data, before you ever look at any of the data, you want to write down one, what is your sample size going to be? How did you determine that sample size? And also, what are the hypotheses you want to test? Like, what's the question you want to ask with this data set? And how are you going to ask that question? What's the statistical test that you're going to do to ask that question? Basically, before you ever do the study, write those down and submit them to a place where they can basically be kind of time-stamped and can be held sort of in escrow, right? So the idea is, like, before we ever analyze the data for this particular MRI QC analysis, we saw that, we wrote down exactly what we wanted to do, and then we went and did it. Can you talk about some of those deviations from registrations and why they don't threaten to inflate the false positive rate? So it really depends on what exactly the deviations are. So for example, if you, I can't actually remember. So, for one study that we published, we had initially said, we're going to do this particular type of dimensionality reduction. And then by the time we actually, had collected the data, we realized that what we had proposed early on was not actually the way that we want to, that it no longer seemed like the right way to analyze the data. We made that decision without actually looking at the data. We just realized that, when we wrote it early on, we didn't quite understand, what all the options were. And we decided that there was a better way to do it. For example, using factor analysis rather than principal component analysis. And so we justified that, right? And I think that, if the decisions are made not based on actually looking at the data, but on some other feature, then I think that there's relatively little concern about inflating the false positive rate. Now, sometimes you're going to look at the data and realize you have to do something differently. So for example, let's say that you collected fMRI data set and you had, proposed to model motion in one particular way, but then you see that the motion turns out to be much worse than you expected it to be, and you need to do more aggressive modeling of motion. Again, I think that, as long as those deviations are made very clear and the motivation for them is clear, then as a reviewer, I generally, obviously it depends on what they are. If the motivation was, well, we did the analysis and there was no significant effect, so then we tried something else, that's, obviously that's problematic. But in many cases, I think one can make a reasonable argument. So the effects of pre-registration are really striking. So this is actually from a very different domain. These are results from clinical trials of treatments for like heart disease and stroke from the U.S. Heart, Lung, and Blood Institute. In the year 2000, they required people to start registering their primary outcome. That's basically, what are you going to measure that you're going to decide is the outcome? Is it going to be, like the number of heart attacks? Is it going to be the number of deaths? And, some change in a biomarker. What is your primary thing that you want to measure? Now, prior to 2000, somebody could do somebody could do a clinical trial, and they might have initially thought they wanted to measure deaths, but then they didn't see any effect on deaths, but they see an effect on number of heart attacks, and so they decide to claim a positive effect there. And so that's why you see before this registration was involved, nearly the majority of clinical trials, these plus signs basically show that the clinical trials are claiming to see a benefit. Year 2000, suddenly you have to say, what is it that you're going to be measuring? No, you can't move the goalpost anymore. And now nearly all clinical trials are showing basically a null effect. Only 2 out of these, however many, 20 or so trials actually showed a positive effect. 1 actually showed a harmful effect. And so this shows that, when in the real world, pre-registration has a substantial effect. Okay, so there's a question, does it make sense to do a pilot study with a few subjects and then use that to create a pipeline for pre-processing the data and then do your main study and apply that pipeline to everyone? Yeah, I think that's a great idea, that having a pilot data set to kind of develop one's processing on and make sure that everything is going to work before applying it to the main data set is great. I mean, I wish we had better tools for creating simulated data sets where you could basically take your real data set and create a knockoff that is kind of statistically similar but doesn't have the same signal, and use that to do your analysis. But we don't have great tools for that yet. I know people are working on it. Let's see, other questions. Could you please elaborate more on how to delineate a clear difference between exploratory and planned analyses? What I mean by that is basically, let's say that you collect an fMRI data set and you wanted to see whether there is a difference in, amygdala activity on a particular task between healthy and depressed people. That's your planned analysis. You would go do that analysis. But let's say that you also think, well, maybe there are differences in connectivity between, amygdala and prefrontal cortex in these different groups. But we didn't write that down beforehand. We didn't put that in our pre-registration. So that would be an exploratory analysis, right? That would be an analysis that you came up with afterwards, and that you didn't have a prior hypothesis for. Trevor is saying, surely there were more clinical trials than that. Are these a subset? I would have to look back at the paper and the Kaplan and Irvin paper and see about that. I'm sure there were, and I don't know how those were filtered. Okay, so another question from Jacob. This is in relation to your power discussion. What role does within subject power play, and can increasing within subject units help alleviate concerns about between subject power? In mixed models, both level 1 and level 2 units contribute to power. Yeah, that's a really good point, that power is determined, 1 by the sample size and 2 by the precision of the estimates. And, there's sort of the interplay of those is going to be relatively complex. But there's some recent papers that I haven't read in detail that sort of start to look at this. So yeah, I think that that a really important thing to worry about is not just the sample size, but the amount of data collected per individual and the precision of the measurement, because that fits in with the reliability of the the measurements. And so there's a recent paper by Elliott et al. That I think was a striking demonstration of how commonly used task fMRI measures that collect relatively little data per individual are actually pretty strikingly unreliable. If you if you do a test and a retest, you see that the correlation between them is relatively low. And so I think that we need to think both about recreasing the amount of data we collect per individual. And I think there's in the last few years, there's been a move towards that. Certainly the recent work from Nico Dozenbach and his colleagues in the Midnight Scan Club has shown that, looking at individuals in much more depth can be a really powerful, way to ask these questions. There's a question, do I think generative adversarial networks can be used to generate usable simulated data? Yeah, I mean, I know people are working on that. I haven't seen any good demonstrations of it yet, but I think that's going to be something that could be used in the future. So David says, one problem I see with pre-registration is it slows down time to publication. Many researchers don't see any incentive to pre-register. I don't actually see it slowing down time to publication. Well, I mean, it takes you time to write down what you're going to do, but I think that, you're going to have to do that writing at some point anyway, right? Either when you write the paper or before that. And I think that, writing down what you're planning to do early on, I think that the cost in terms of time to publication is pretty low. The bigger cost is the bigger cost is really I think in the fact that you're going to have many more you're going to have many fewer positive outcomes if you're pre-registering, right? Because as we see here, you have a lot more null outcomes, and given that null results are harder to publish, it's going to make it harder on you. But it has the benefit of making you not publish false stuff. And I think most of us don't want to publish false stuff. Are ICA network results reliable? It depends on what you mean by reliable. But in general, yeah, I think that because they're collapsing across more data, there's a decent reliability of those things, but there's a lot of details we could get into. Okay, there's a question about multiverse. I'm gonna move on and come back to the multiverse question in a little bit, because that's actually something I'll bring up at the end of the next section. Okay, thanks for all the great questions. Keep them coming. So one point to make about pre-registration is that it would prevent things like p-hacking, but it doesn't eliminate analytical variability, right? Different people are still going to have different workflows, and so we wanted to ask, how variable are workflows in the wild? If you actually look at what people are doing in their labs, Josh Karp kind of looked at this in his paper, right? But the question is, like, what is the real world effect on scientific inferences? And so we put together a study with a number of behavioral economists, and this is in collaboration with Tom Schoenberg's group in Tel Aviv, and Tom Nichols, looking at basically what happens when you give people a data set and ask them to analyze it. So we collected a data set based on a previous study that we had published a while ago, 108 subjects performing a decision-making task, and then basically we recruited teams to analyze the data set, up to 3 people each. And then we had them sign a non-disclosure agreement so they wouldn't talk to anybody about what they found until we lifted the embargo. We gave out the data and gave them 3 months roughly to give us a set of answers to 9 questions, which are of the form: is there significant activation in some particular brain area for some particular feature of the experimental design? And we told them to basically, use their standard analysis workflow. The only thing we said is you need to do a whole brain analysis. We got results back from 70 teams. And in addition to their yes/no decisions, we also got their whole brain statistical maps, both before and after thresholding. And so the first thing to note is that, they gave us detailed descriptions of their analysis workflows. None of the 2 teams used an identical workflow. And even with these detailed analyses, these detailed written descriptions, it was often hard to tell exactly what they had done. So here's the kind of the main finding of the study. So what I'm showing you here is the proportion of teams reporting a significant positive effect for each of the hypotheses, ordered in terms of those proportions. So basically, let's think about what would happen if people were perfectly reliable. So either they would be reporting 0, that is, everybody would say no, or this would be 1, that is, everybody would say yes. What we see is that for 3 of the hypotheses, basically we're pretty close to what you would expect if there really is no true effect and everybody was not finding it, right? For this one hypothesis, the majority of teams, about 85% of teams, said yes, there is an effect. For the others, they were kind of in the middle, right? So 20 to 40% of teams were saying, yes, there's an effect, right? And so another way to look at it is, how many different patterns of outcomes were they? If you look at the 9 yes/no outcomes, there are actually 33 different patterns of outcomes across those 70 teams. And for any particular hypothesis, you can find at least 4 workflows that can give you a positive result. This is what happens if you look at the whole brain results. This is the proportion of teams reporting a positive significant activity in each voxel, right? And basically what you see is that the voxel with the highest overlap, it was only found to have activity by 76% of the teams, right? So there's no voxel where everybody is saying, yes, there's activity. There's a lot where everybody's saying, no, there's no activity. Um, and this is another way to look at it. Like for each hypothesis, if you look across teams, what's either the minimum or the maximum number of significant voxels that were reported? And basically for each of them, the minimum, so, there's always a team that basically says, no, there's no significant voxels for this particular thing. But then you could have another team that's reporting 118,000, 135,000 voxels, right? So this just shows you how variable those results are, across teams. Now it turns out that the underlying, if you look at the whole brain statistical maps, they actually, were fairly similar across a lot of the teams. So there's this is a correlation matrix of the whole brain Z statistics, the unthresholded statistical maps. And this is basically showing there's a big set of groups here that, are all reasonably correlated with one another. Even though the red and the green up at the top shows basically for this particular hypothesis, whether they said yes or no. And you see that even for teams that have pretty similar underlying activity, they can disagree on whether there's actually, activity in this particular region or not. We also found a number of teams that were actually anti-correlated with the main group. And it turns out that these teams had basically misspecified the model for ways that I'm not going to have time to go into. And there's another set of teams that were kind of uncorrelated with everybody else. And we didn't dig deeply into what they had done, but we're assuming that they probably made some kind of mistakes as well. On the other hand, if you do a meta-analysis across, the various teams, the unthresholded maps, you do an image-based meta-analysis, you actually see that there are consistent results. So for example, this result right here for hypothesis 5, this is the one that, that had the 86% agreement. You see that there's a number of regions that actually show significant, effects. So it's not as if people aren't finding consistent things. They weren't finding consistent things for those particular hypotheses, but the underlying statistical maps actually do show some consistent results. So this is what leads us to think that, and this is in response, this is related to the question that was asked about moving towards what we call multiverse analyses, right? And the idea here is that for any particular data set, other people have worked on this type of analysis in other areas. They sometimes call it sensitivity analysis. The idea is basically, John Ioannidis calls it vibration analysis. The idea is like, how much do your results change when you analyze the data differently? So what we think, we want to move towards is actually for any particular data set, run it through, a number of different pipelines, and then look at what's convergent across those pipelines. And obviously this is computationally challenging given the size and complexity of fMRI data sets. So members of my group have been working on a project called FitLens, which basically tries to automate this using something that Tal Yarkoni has developed called the BID statistical models framework, which basically provides you with a machine readable way to describe a statistical model for fMRI. And that's kind of work that's ongoing right now. Okay, so I'm going to pivot now to talk about another set of issues. Maybe I'll take just a moment to see if there's any other questions that people want to type in. Okay, could you talk about how to do power analysis for neuroimaging studies? Yeah, power analysis is just a hairball. It requires so many assumptions and
Russ, can I just mention that JB will show some mechanics of that.
Okay, good.
With code during his talk on Wednesday, I believe.
Okay, so I'm actually going to leave that to JB to say more about then. We've developed some tools, like we have a thing called NeuroPower that you can find online that helps with this, but it requires a ton of assumptions. I think JB will tell you more about that. Okay, any other questions? Okay. Is the multiverse analysis currently specific only to fMRI data, or can it be applied to DMRI data, for example? Right now, the FitLens tool is built for fitting linear models to fMRI data. If you were trying to do some kind of linear model on diffusion data, then you might be able to do that. But for example, if you wanted to run a bunch of different tractography algorithms or something like that, that's going to require a different setup. And I don't know if Ariel has thoughts about that.
not many thoughts, except that it would be a great idea to do that.
Yeah, right. Okay. For multiverse sensitivity analysis, how many variations of the pipeline should one conduct? I don't know that we have a good answer for that yet. I think we need to, and we're working right now on actually starting to do some multiverse analysis on that NARPS data set. So I think we'll have a better idea. My guess is, tens or hundreds. In general, when people do sensitivity analysis, it's, the numbers are relatively large, and obviously that starts to get really computationally expensive. But I think that that it's probably going to be on that order. Okay, cool. So I want to move now to talk a bit more about reproducibility in the context of coding. Obviously, you're all here because you want to learn to kind of do code your own analyses, and I think there's a set of things that we have to think about and worry about when we start doing that. So if I was in the room with you, I would ask you to hold up your hands and ask, how many of you have written computer code in the course of your research? Almost all of you would raise your hand. How many of you have been trained in software engineering? Some of you might raise your hand. How many of you have ever written a test for your code? A few of you might raise your hand. And so I want to, point out just, what the risk is of basically using code written by scientists rather than software engineers. This is Jeffrey Chang, who is a structural biologist, who published a bunch of papers in the early 2000s about this particular protein called the ATP binding cassette. He's a structural biologist, so they try to use X-ray crystallography to understand the structure of proteins. He had these 3 science papers, all of which had to be retracted because somebody else realized that his structure was wrong. And when they dug in, it turned out that there was a piece of software they were using that they'd gotten from some other group that basically introduced a sign change at one point in the analysis and caused basically them to come up with the wrong structure. And so, this is just one example of where this has happened. I wrote a blog post a few years ago about, an example of how a tiny error that I had made had a pretty big effect. We were working on a paper. This was looking at classification of cognitive tasks using fMRI data. So basically, we have individuals doing one of 23 different tasks. And I wanted to ask, can we tell which task you're doing based on your brain activity when I haven't seen your brain before, using data from other people. So I put it into my, scikit-learn model, and I got out 93% accuracy. Now, if I had gotten 100% accuracy, I would have known I've done something wrong. And 93% accuracy is pretty amazing here, right? Because, this is 1 out of 23. So chance is down at 3%, 4%. And so, clearly, something might have been wrong here, but it didn't. 93% just seemed amazing, right? So we were working on the paper, and a grad student working with me, Sammy Koijo, who's now he became a postdoc and now faculty at Illinois. He said, "Oh no, you messed up. You left out this index on your array right here." So I just left out 2 characters right there. And basically meant that I was indexing the array in the wrong direction. So I was classifying over voxels rather than people, classifying over people rather than tasks, I think. And so when I did it right, it turns out that it went down to 53% accuracy, still way above chance, but much lower than that 93%, all due to that one tiny little mistake that, that I never noticed. Now, a real concern here is, what I and others have started to call bug hacking, which is the idea that, if we have a bug that confirms our predictions, we're much less likely to find it than if it's a bug that disconfirms them, that we don't like, right? And that's kind of, highlighted in this PhD comics thing, right? Did it confirm our hypothesis? Yes, then stop checking it. So I have a principle that I wrote about in another blog post a few years ago that I call the principle of assumed error, which is basically whenever we find a really good result, we should assume that it occurred due to an error in our code rather than, due to us being right. So the idea is that, our prior should always be that we've done something wrong, and we should work really hard to try to figure out what we might have done wrong. And, an example from my lab is, we had a paper we were writing up, and one of my postdocs just thought something couldn't be right about the result. He spent about a month digging through all of the code that led to that result, and finally found the bug that that turns out invalidated all of the findings, where some trials had been misclassified, I think, and had we not done that, that paper would have been published. And so I think it's really important to kind of work as hard as you can to figure out how you might be fooling yourself. And one of the things that we can certainly do is software testing. And so there's lots of different types of tests. I don't know if will somebody talk about testing later in the week?
I will talk during the Python packaging.
Great. Okay, cool. So you'll hear about lots of different types of tests, right? Some of them are just basically, does everything run without exploding? Others are, making sure that the numbers are in reasonable ranges. Some of them are, if you have a function, making sure that that function actually does what it's supposed to do. So you'll learn more about that later on. Here's an example where, a test would have saved some researchers a lot of pain. So this group had published a paper about, the role of conspiracy theory and in rejection of science. And they had initially said that, there was no relationship between age and several of the things that they were interested in. But it turned out that when somebody looked at the data set that, these were supposed to be data from adults, but they had one subject reported with age 5, another subject reported with age 32,757. And when they fixed that, it turns out, well, now age does correlate with these other things. And so that sort of was a problem for the claims that they were making, because it was a confound. Had they simply, made sure that they were testing to see that their ages were all within reasonable ranges for age in their data set, this would have protected them from making that error and having to make a correction to their paper. So one of the things that I think is really useful and important is kind of spending some time learning about software engineering. I don't know that going and taking classes is obviously, different people have different ways that they try to learn things. I found these 3 books to be particularly useful in kind of, laying out lots of ways to think about writing better code. And so I would definitely, recommend all of these. None of them is light reading, but they're all like really useful for like learning. Writing code is a craft, right? It's like, or a skill. It's just like learning a new spoken language or playing a musical instrument. You have to do it a lot. You have to work at it, and you have to intently work at it, like trying to get better, right? And so so I would suggest spending some time, if writing code is something you're going to spend a lot of time doing in your science, you want to get as good at it as you can. So one of the tools that you might learn about is a set of tools that let you perform automated testing. So the idea is, if you hook these up to your GitHub repository, now there's a new one called GitHub Actions that makes it really easy. The idea is that, if you have a set of tests that you've written, they'll automatically get run whenever you push a new commit into your GitHub. And so you'll always know whether you've kind of broken anything. Another thing that I think is a good improvement to one's coding practices is code quality analysis, and both in terms of like looking for errors and also adhering to style, right? So it turns out that you don't have to adhere to a particular, you don't have to write your code in a particular style, but doing so makes it much easier for you to know what you've done in the future and for your collaborators. So I spent time last summer when I was writing code for the NARPS project. Basically, I spent a couple of weeks going through and basically rewriting all the code to adhere to the Python. There's an official Python style guideline called PEP 8. And so I basically went and rewrote my code using tools that can sort of automatically tell you, whether your code meets the style guidelines. And then I had, I was working on code review with another colleague, and they basically said, fixing your style made your code so much easier to read. Another thing that I think is a good improvement to one's coding practices is using, whenever possible, using an established library. So there's, a lot of people, especially people coming from engineering backgrounds, like, think that the only good solution is the one that I've written, right? And so even if there's 20, well-engineered solutions out there, they might have the inclination to try to write something their own. So for example, the canonical example I use is somebody who thinks they need to write a new DICOM to NIFTY converter. Now there sometimes might be a good argument for that, but in general, it's always better to, if there's a tool out there that does most of what you need, take that existing tool and try to contribute a fix or an extension to it, rather than trying to write your own from scratch. And in particular, try to use libraries that employ good software engineering practices like testing. So for scikit-learn being a good example of that. So I want to spend the last few minutes talking about basically what I did in developing the analyses for the NARPS project. So we took, we got the data from each of the analysis teams. We got their hypothesis results, we got their maps, and we wanted to build a kind of a fully reproducible analysis workflow. And there's a question about what kinds of checks might identify issues in our code. I'll come back to those in a little bit. So I wanted to basically write an analysis pipeline that could take the raw data that people could obtain and fully reproduce everything up through the figures that go into the paper. So the first thing that requires is all the data be available and versioned, right? So on OpenNeuro, when you upload a data set, you have to create before you share it, you have to create what's called a snapshot, and that snapshot becomes immutable, right? So if I tell people I was working with version 1.0.4 of the data, they can grab exactly that data set and know exactly what I was working with. Sort of, as perfectly as possible. And so we wrote a data paper. It has a link to this digital object identifier where one can grab exactly the data set that we used. And I'm not going to say much about BIDS. BIDS is the format in which those data are shared, and it makes it easy for anybody to take the data and work with them pretty easily. So we had very little back and forth with the people who took the data from us, because BIDS made it really clear, what exactly was in those data. You're going to learn more from Chris Gorgolewski about Docker and containers. And there's a great quote from David Donahoe from a few years ago. An article about a computational result is advertising, not scholarship. The actual scholarship is the full software environment code and data. So we talk a lot about, sharing of code. We talk about sharing of data. Containers give us a way to share that entire platform. And we know now that, different versions of software libraries and software packages can result in substantially different results. And so containerization allows us to provide people with an exact replica of the computational platform that we ran things on. That doesn't mean that it's perfectly correct, but it's at least perfectly reproducible. Now, in Python, it's nice because you can tell, when you install software, you can tell pip to install exact versions of individual packages, like NIBAB will use 2.4.1. So then we can make sure that we're doing that. If you work in R, which some of our analyses here were in R, that's a lot trickier because it's pretty much impossible to tell it, especially for dependencies, exactly which versions of libraries to use. But there is a cool package from Microsoft called Checkpoint, which basically fixes this problem by letting you specify a date and saying, give me all the libraries that were current as of that particular date. And so this is what we did for NARPS. We basically said that we pinned it to July 16, 2019. And so that way, anybody can now go back and reinstall all those same packages because they're not going to change at whatever was current as of that date. Another thing that if you don't already use makefiles, you should learn how to use makefiles. They're super handy, both for you as a developer and for users. And if you go to my GitHub that I pointed to back there, you can see that. This is basically showing what it takes to set up an automated test. And what I actually did for NARPS was have the tests actually run all of my analyses. So I basically, for every analysis, I created a test that would run that complete analysis and save all the results. And then it turns out that using this, I was using Circle CI. I think GitHub Actions is probably the easiest thing to use these days for automated testing. But the idea here was that anytime I made a change to the code, that change I push it to GitHub, it would trigger this Circle CI thing to automatically run all of my code. It took, ultimately it took about an hour and a half, I think, to run all the analyses. And then all the data, the images, everything, all the results were saved out to a file that I could then basically take and share. We also implemented some automated code quality analysis. There's a tool called Codicy that you can use that basically will run a bunch of checks and ultimately give you a letter grade as to how good your code is. A lot of it is stuff that doesn't matter that much for our analyses, like security stuff, but it can also tell you, for example, are you doing something that's error prone? Now, I think there's no substitute for having another human look over your code. And so one of the things that I did was I got Tristan Glatard. I don't know if Tristan's going to be there for this year's NeuroHackademy. But I got him to basically go through all of my code after I rewrote it to match the Python style. And basically, one, ask, am I making any logical mistakes? Am I doing anything that smells wrong? And also just looking for kind of stupid little mistakes. He found a bunch of different issues. I fixed them, and it definitely helped the quality of the code a lot. Another thing that's really common. So code review is something that people in industry do all the time. Now, you don't always have somebody who's a highly expert coder to review your code. One thing that I think could be useful for novel coders is basically working together with someone to write code, what's called pair coding, where you and another person look at the same code at the same time and basically write it together. You're not going to be sitting in the same room right now and doing that, but there are a lot of virtual tools. So for example, if you use Visual Studio Code, it has a way to basically do collaborative coding, and you can have a chat going on at the same time, a video chat going on at the same time that you're coding. So it's as close as possible to being in the same room. So getting to somebody asked this question about how can you write tests that can tell whether you've done something wrong, right? And so this gets to this distinction between reliability and validity, right? A result can be fully reproducible, but still be wrong, right? If I fill all of my, all of my data with the number 0, the results are going to be perfectly reproducible, but they're going to be wrong. So there's a couple of things that we like to do for complex analyses. One is, whenever possible, and this is sometimes difficult, generate data where you know the true answer, and then feed that into your code and make sure that your code can get back the right answer. Then you can also do the flip side, which is basically generating data where you've basically generate random data where you know that the null hypothesis should be false, should be true, right? And then run that a bunch of times and make sure that your false positive rate comes out where it should, right? If you do 1,000 analyses, 5% of those should be significant if you're controlling your false positive rate at 5%. This is basically what Anders Eklund did in his 2016 cluster failure paper. Obviously, it's best if you do this in advance of any real analyses and you pre-register it. So what we did for NARPS was basically we took the known data, generated simulated data based on those known maps, included different levels of variance, included some kind of real features of the data, reran the entire analysis stream on the simulated data, and then basically asked, what's the correlation between the final analysis and the ground truth maps that were used to generate the data? And basically, if that, I consider that assessed, like if those are correlated at some high level, like greater than 0.95, then I said, okay, look, we've done a good job of recreating the results. Obviously, there's some noise, so it's never going to be perfect, but they should be really close. So let me, I think that's that's about all I have. So let me just conclude, and then I'll kind of turn to questions. So if you have other questions, go ahead and start writing them in. So, I hope I've convinced you that we have a reproducibility problem. I don't think it's any different from other areas of science that have like similarly large data sets and complex computational workflows. I think one key to solving the problem is statistical power. I think another is, basically improving our software. And I think, particularly NeuroHackademy is a place where you're going to learn a lot of things that can help you write better code. I think that, kind of continuing to educate yourself about tools and practices of software engineering is really important. And I know that, you're all going to walk away from NeuroHackademy knowing a lot more how to do this stuff. Let me just thank all the people who've been involved, and also thank Chris Gorlewski, who helped kind of start the center at Stanford and now has moved on to Google, but you're going to be hearing more from him. Okay, so let me jump into some questions. Okay, preparing to do my first fMRI analysis, therefore learning as I go. I want to register hypothesis to OSF and worry about leaving out important details. I think that's always a challenge, knowing like what are the right details to include. I think a philosophy that I have is that you shouldn't let the perfect be the enemy of the good, right? That doing something is better than doing nothing. So even if you just write down, this is my anatomical region of interest, this is the contrast that I want to test something on, or this is the particular question that I want to ask, this is what my sample size is going to be, this is what my statistical threshold is going to be. Even writing down those few things, even if you don't write down, this is my entire workflow, this is exactly how I'm going to model things, I think that it's still better than doing nothing. Obviously, the more detail, the better. And the reviewers are going to, sort of want as much detail as you can give. But I think doing something is better than doing nothing. And I've not seen examples of people being penalized for, for not giving enough detail. Okay, let's look at some questions. If one needs to collect a new set of data because there's no available data, and one has to choose between a large number of scans, a small number of participants versus a small number of scans, and a large number of participants, what is the better way to go about that? So I don't think there's a single answer to that, in part because it depends on the kind of question that you're going to ask. So, there are certainly literatures where people regularly collect, a very small number of individuals with very large amount of data, and particularly areas like vision research, you'll often see papers there with 4 subjects where, you've collected a ton of data on each individual and really mapped out, the response of those individuals. I think that kind of approach is often reasonable, especially if you're not looking to make claims about individual variability. I think the place where you really need large number of individuals is if you want to either ask questions about individual differences or about group differences. And I think those are the places where I see the reproducibility problems in our literature being the worst. And that's why I highlighted, both the irreproducible correlation study and the depression study, because I think those are the areas where we have a lot of concerns. The other thing you can do is, and I think looking to the Midnight Scam Club papers, is a really nice example of this, where, if you characterize individuals in detail, you can basically present the data as like a set of case studies. So there, you're not trying to do group statistics. You're not trying to do a t-test across 10 people. You're basically saying, hey, look, here's what we find for each individual person. And so I think that's another, it really depends on the kinds of questions that you're trying to ask. Okay. Are there some other ecosystems programming languages besides the one around Python for neuroscience development that include area specific error checking? I can't, I know about Python and R primarily, and R definitely is much weaker in terms of its kind of, software engineering ecosystem. I can't really speak to others. So that might be a question to raise with other people later in the week. Do you have recommendations about how to implement robust testing on a platform like CircleCI or Travis without disclosing PHI? So yeah, this is challenging, depending on the nature of your analyses. So, I think the first thing to say is, a lot of the testing that you might do in, it doesn't necessarily require real data, right? So I think a good way to go about that is to basically have your tests run on kind of, data that you synthetically generate. And they could be random data, they could be realistic data. But if you have, data that you can use for your tests, or you're running tests that are more granular that don't require kind of, full data sets, then you're not going to need to upload anything with PHI. Yeah. Now for the NARP study, there was, because we were using the group statistical maps, there was no PHI, involved in those. And so, so that's, those are some options. Okay, so there's a question, is it worth pre-registering and trying to submit? Oh, where'd the question go? Oh, it disappeared. Okay, so must have been answered. So how do you think hyperparameter tuning and cross-validation relate to the multiverse approach? I mean, I would see those as living, I certainly think it could make sense to ask, within a multiverse sort of approach, how do a bunch of different cross-validation schemes relate to one another in terms of the accuracy that you get out of them? So there's no reason that multiverse couldn't be applied to, questions about, predictive accuracy as opposed to questions about statistical hypothesis testing. And so, I certainly think that that's a very legitimate thing to do. I haven't seen a lot of examples of that, but I think it's a reasonable thing to do. Many people I've talked to are worried about false negatives, basically that the methods we use to control our false positive rate are too conservative. Have you heard this from people? How do you think about this, both as a real problem, but as a perception? So I've certainly heard that from people. Yeah, there are papers written. I think Will Cunningham and Matt Lieberman had a paper a few years ago trying to make this argument. Really the question is, what's the cost of a false positive versus a cost of a false negative, right? And those are going to be different in different domains, right? So if I'm a surgeon and I'm trying to use fMRI to decide, like, is it okay to cut out a piece of brain? And I'm using, because I don't want to cut out like language cortex. The cost of a false negative is pretty high, right? So there you really want to make sure that you have enough power to find things, and you want to kind of penalize false negatives really heavily. I think the reason that many of us worry a lot more about false positives and false negatives is simply that false positive, once an effect is like put out there, that it can have, it can basically live on for a long time. We know that, even once an effect has been sort of debunked, people continue to cite it for a long time. And so the concern is that people, if there's a false positive, people will try to build on it. And so I've, for example, I've had students in my lab spend years trying to, trying to basically follow up on a published finding. And ultimately after, 2 or 3 years, realizing that published finding is not reproducible. And then later hearing from other groups who also had failed to replicate it, but hadn't actually, published that failed replication. And so that false positive led to a student spending a lot of time. Actually, one of my students left science based on such an experience, because it was just, so frustrating. Whereas, I think false negatives will often, could potentially cause people not to look at something. But, often, somebody might think if it's a false negative, there's a lot of reasons that one can have a negative finding. And, I think it's not going to have the future impact of a false positive. That's for me why I think, in scientific publication, especially for like, the kind of fMRI studies that we do, like healthy adult studies or group different studies, errors of commission, false positives, I think have a much more a negative effect on the literature than errors of omission or false negatives. But obviously different people might think about this differently. What are your thoughts on Bayesian?
Russ,
sorry?
I'm gonna cut this off now because we have another live session starting in 10 minutes. So maybe we could take the question about Bayesian analysis on Slack instead and continue the discussion there. thank you so much, and see
All
you
right,
all
thank you
soon.
all.
Bye.